The gut-brain axis is, without question, one of the most productive and surprising frontiers in contemporary neuroscience, and fecal microbiome transplantation as a psychiatric intervention is its most counterintuitive implication. The basic concept that transferring gut bacteria from one individual to another can influence brain function and mental health sounds, on first encounter, implausible enough to dismiss. The animal research makes that dismissal very difficult to sustain. The foundational experiments are compelling. When researchers transplanted gut bacteria from anxious mice into germ-free mice, animals raised without any gut microbiome, the recipient mice began displaying anxiety behaviors. When the transplant came from calm donors, the recipients became calmer. This transfer of behavioral phenotype through gut bacteria alone is one of the more extraordinary findings in recent neuroscience because it demonstrates that the microbiome is not merely correlated with mood and behavior. It appears to causally influence it. The mechanism is becoming clearer. The vagus nerve, which runs from the brainstem directly to the gut, functions as a bidirectional communication highway. Gut bacteria produce neurotransmitter precursors. Most notably, approximately 90% of the body's serotonin is produced in the gut, not in the brain. The gut microbiome also regulates local and systemic inflammation, and chronic neuroinflammation is increasingly implicated in depression, anxiety, and cognitive decline. When the microbiome is disrupted, these downstream effects ripple through the nervous system in ways that are only beginning to be mapped. Early human trials are producing signals, not conclusions, but signals of neurological improvement following FMT in autism spectrum disorder, in depression, and in multiple sclerosis. These are preliminary findings requiring replication and much larger trials. But the signals align with the mechanistic story in a way that justifies serious scientific investment. There are significant regulatory and safety considerations that deserve directness. FMT is not a benign procedure. Donor screening is critical. There have been deaths from FMT procedures in immunocompromised patients who received transplants from donors carrying drug-resistant pathogens that screening failed to detect. The regulatory status of FMT sits in a genuinely awkward position. It is not a drug, not a tissue transplant in the traditional sense, and not a living biological product by existing definitions. It is, effectively, a new category that regulatory frameworks are struggling to accommodate. The field is moving. The science is genuinely exciting. But the jump from treating C. difficile infection to treating depression or autism should not be made carelessly. Each new indication requires rigorous trial design, safety protocols, and honest communication of what is known and what is not. The gut-brain axis research is real. The therapeutic applications are promising. The clinical infrastructure needs to develop at the same pace as the scientific enthusiasm, rather than trailing behind it.